Google has introduced DeepSomatic, an advanced AI tool capable of identifying cancer-related mutations in tumor genetic sequences with superior accuracy compared to existing methods. This development, published in Nature Biotechnology, marks a significant step forward in cancer research and the realization of precision medicine. The critical need for precision. Cancer starts when the mechanisms controlling cell division malfunction. To create effective treatment plans, it is essential to find the specific genetic mutations that are driving a tumor's growth. Doctors regularly sequence the genomes of tumor cells obtained from biopsies to inform treatments that can target how that particular cancer spreads and grows. However, cancer genetics is highly complex. While sequencing finds variants, distinguishing real genetic variations from sequencing errors is extremely difficult to challenge where AI tools provide welcome assistance. The challenge of somatic variants. Most cancers are driven by somatic variants, which are acquired after birth rather than inherited germline variants passed down from parents. Somatic mutations occur either through environmental factors, such as UV light damaging DNA, or via random errors during DNA replication. If these variants alter normal cell behavior, they can cause uncontrolled replication, thus driving the development and progression of cancer. Identifying somatic variants is particularly challenging because they can exist at low frequencies within tumor cells, sometimes at rates even lower than the sequencing error rate itself. How DeepSomatic leverages AI. DeepSomatic utilizes convolutional neural networks to identify genetic variants in tumor cells. The process often starts in a clinical setting where scientists sequence both tumor cells from a biopsy and normal cells from the patient. 1. Image conversion. The model converts raw genetic sequencing data from both the tumor and normal samples into images. These images represent various data points, including the sequencing data and its alignment along the chromosome. 2. Analysis and differentiation. The convolutional neural network analyzes these images. It works to differentiate between three elements: the standard reference genome, the individual's normal inherited variants, and the cancer-causing somatic variants, all while filtering out sequencing errors. 3. Output. The result is a list of cancer-related mutations. Critically, DeepSomatic is also able to work in a tumor-only mode if normal cell samples are unavailable. This frequent challenge, often encountered with blood cancers like leukemia, makes the tool applicable across many clinical and research scenarios. Training and performance excellence. Developing an accurate AI model requires high-quality data. For this project, Google and its partners at the UC Santa Cruz Genomics Institute and the National Cancer Institute created a benchmark dataset called CASTLE. This dataset was built by sequencing tumor and normal cells from 2 lung cancer samples and 4 breast cancer samples. To ensure accuracy, these samples were analyzed using 3 leading sequencing platforms, combining the outputs and removing platform-specific errors to create a single, accurate reference dataset. DeepSomatic demonstrated superior performance compared to other established methods across all 3 major sequencing platforms. Complex mutations. The tool particularly excelled at identifying complex mutations known as insertions and deletions (indels). Accuracy scores. For indels on Illumina sequencing data, DeepSomatic achieved a 90% F1 score, significantly better than the 80% achieved by the next best method. The improvement was even more pronounced on Pacific Biosciences data, where DeepSomatic scored over 80%, while the next best tool scored less than 50%. Furthermore, the tool showed utility even when analyzing challenging or lower quality samples. Advancing precision medicine. The data generated during the training process revealed how even the same cancer type can possess vastly different mutational signatures, information that can aid in predicting a patient's response to specific treatments. The AI tool has also shown its ability to apply its learning to new cancer types it was not specifically trained on. For instance, it successfully pinpointed the few known variants driving the disease when used to analyze a glioblastoma sample, an aggressive brain cancer. In a partnership with Children's Mercy in Kansas City, it analyzed 8 pediatric leukemia samples using the tumor-only mode, successfully finding the previously known variants while also identifying 10 new ones. Google hopes that clinicians and research labs will adopt DeepSomatic to gain a better understanding of individual tumors. By detecting known cancer variants, the tool can help guide choices for existing treatments. By identifying new variants, it could potentially lead to the development of new therapies. The ultimate goal is to advance precision medicine and deliver more effective treatments to patients. To facilitate this, Google has made both the DeepSomatic tool and the high-quality training dataset openly available.